methods of cross-pollinated crops. We'll discuss on population improvement
principles of plant breeding. Today's topic is on breeding methods of cross-pollinated crops. We'll discuss on population improvement methods and synthetic varieties. By the end of this class, you'll be able to answer the following questions. What are breeding methods of cross-pollinated crops? What are GCA and SCA? What is population improvement and what are population improvement methods? And what are synthetic and composite varieties? If we classify the breeding method of cross-pollinated crops, it can be done in two. First one is population improvement methods and second one is hybrid and synthetic varieties. Well, in the first one, it is basically based on increasing the number of desirable alleles in a population. So if we have a population with decreased variations or lack of variation, so what we have to do, we are going to allow random pollination in the population and we are going to select in the next generation, which we have done in mass selection. Okay, so in the second one we have hybrid varieties, which is formation of F1 hybrid heterosis by crossing two diverse parents. While in this synthetic varieties, we are going to select multiple parental lines and we are going to form a composite variety. So synthetic variety is called a composite variety, not an individual variety. Okay, so population improvement method can further be classified into intra-population improvement and interpopulation improvement. In intra-population improvement, it is based on improvement within the population, which we have already done in mass selection, which is based on phenotype of an individual and composition of the phenotypically superior plants and growing in the next generation. While in case of family selection, which is the second type of intrapopulation, it is based on evaluation of progenies of what? Progenies of a cross formed by in several manners. Okay, so it can be classified into full sheep progenies, half sheep progenies and self progenies. So what is full sheep, half sheep and inbred? Let's see. In half sheep progenies, it is a progeny formed by crossing parents with one of the parent in common. Okay, so here we have A parent cross with B, C and D. So A is common in all this. So progenies which are having one parent in common is called half sheep progenies. Okay, so selection of superior plants from this progeny is called half sheep progeny selection. So in case of the second one, full sheep family selection, so usually this happens this is done with selection of superior several lines like 5 or 6 lines and then crossing as a female and some of the lines as male parents. Okay, if it is cross-pollinated crop plants. So when we have this, so A will be crossed as A, which sometimes will produce into self. Okay, and A can also be crossed as to the second parent, which can form into AB. Okay, and B can also be crossed again into B can cross with A. Okay, so this cross usually is this progeny is usually a cross between A and B, which is all the same with B and A, but the performance may be different because if in certain cases if there is reciprocal differences, okay, then their performance will be different. So selection of individuals based on such type of differential reciprocal difference individual is called full sheep family selections. Okay, so in this case selection is based on progenies which are formed by crossing the same line, several lines, some of acting as female parent and male parents. Okay, so in the biparental manner. While in case of the third one, which is inbred family selection, so in this case it is also same. So we are going to select the inbred line and form the inbreds. So selection of inbred progenies formed by inbreeding is called inbred family selections. Okay, so in the second one, which is interpopulation improvement, it is improvement of two different populations by with the help of one another. Okay, so if you take a reciprocal recurrent selection, which we are going to discuss later on, so in that case both populations A and B act as a tester for one another. Okay. So before we actually move on to the population improvement methods, we are going to study about some important concepts. First one is combining ability. Okay, so what is combining ability? It is the capability of an individual parent to transfer its traits into their offspring. Okay, and it also means the compatibility between two parents and fruitfulness of the F1 hybrid form from the crossing two parents. Okay, and which usually means hybrid vigor itself, or you can call it as heterosis. And progeny tests should be performed. Okay, what are progeny tests? We have already witnessed here half shift progeny test, full shift progeny test. Okay, for estimating the combining ability, progeny test should be done, and especially for characters which are governed by recessive genes. And what are the different types of combining ability? We have general combining ability and specific combining ability. So coming to general combining ability, it is the average performance of a line, say for example A here, when crossed with several other lines B, C and D here. So it is the average performance of crossing of a particular parent with several other parents. Okay, so it is usually, the general combining ability is usually associated with high additive genetic variance and additive genetic variance. Okay, and when this additive genetic variance is high, it usually means it is suitable for selection. Okay, it is recommended for selection, and it denotes combining ability of a genotype, especially with inbreds or with the various testers. Okay, and inbred with various testers. It helps in identification and selection of best genotype to use in hybridization as a parent. Okay, not only selection, it indicates general combining ability indicates the selection of a type of parent, criteria for a parent to be as a parent in hybridization program. It is estimated by half-shift mating, as we have already seen what is half-shift mating, and we have and they have a relationship with narrow sense heritability. What is narrow sense heritability? It is the ratio of additive genetic variance by total phenotypic variance. So as we have already seen here, additive genetic variance is directly correlated to general combining ability. Next is specific combining ability. It is the performance of a parent under consideration in a specific cross, say especially in case of biparental cross as such. So performance of a specific cross is usually tested by testing it against their several reciprocal crosses. Okay, it is a deviation from general combining ability. And according to general combining ability, it is the average performance of a particular inbred. While in case of specific combining ability, it is the combining ability of that particular cross. Say for example A cross with B, not B cross with A. So it is, or B cross with C. Okay, so it is the combining ability of this particular cross, okay, from others, and it is due to dominance genetic variance. So whenever dominance genetic variance is high, it is usually recommended for hybridization. Okay, and it helps in identification and hence selection of best cross combinations, that is those with desirable outputs. When we see that an inbred line combines well in any cross, it is due to specific combining ability. So this is very much important for selection of a parent as during a hybridization program. If high specific combining ability is there, you can then select these parents for uh, making a cross. Okay, it is done by analysis of specific combining ability is done by full shift mating, also known as biparental design. Okay, and they have relationship with heterosis. Okay, so the important point to be noted here is GCA and SCA is important while making a hybrid variety, F1 hybrid variety, to get heterosis. So before conducting a hybridization program, first we have to make sure that the GCA and SCA is high for that particular cross, and we have to conduct a hybridization program later on. And especially for synthetic varieties, apart from hybrid varieties, synthetic varieties also there. So we have to, in synthetic varieties, we have to consider only the general combining abilities of each and every component lines, okay, which will witness later. Okay, so coming to the population improvement method, recurrent selection. So this comes under in intrapopulation method. So recurrent selection is basically mean it is the reselection of generation after intermating. Okay, so this is very simple ideology where intermating is done and reselection in the next generation, or it can be done once. Population improvement can be done once, or it can be repeated several times until and unless you gain desired genetic variations. So it says here that reselection of plants generation after generation with interbreeding of selected plants to provide better genetic recombination. It is usually described by Jenkins in 1940. The term recurrent selection was given by Hull in 1945. Recurrent selection is efficient breeding method for increasing the frequency of superior genes in a population. So these all are important for competitive exams. So again here the recurrent selection can be divided into 4 different types. Simple recurrent selection for general combining ability, improving the general combining ability, recurrent selection for improving specific combining ability, and reciprocal recurrent selection. So let's see one by one what are those. In simple recurrent selection, selection is based on phenotypic characters of plants, and there is no tester used here, and it does not measure the combining ability. Okay, GC and SCA is not measured here. It is only for those characters which have high heritability. Okay, for those characters which is usually governed by recessive genes or recessive alleles, it is not recommended. Those are recommended for recurrent selection for specific combining ability and general combining ability. So in this simple recurrent selection, we have a base population. So from a base population, we are going to select superior phenotype, and we are going to self the selected population, selected lines, and harvest the seed separately. Okay, so the lines which are indicated by red is the superior lines, and we are going to select from this base population, and we are going to harvest separately. And the harvested seeds are planted in individual plant progenies. Okay, so these selected lines are planted in individual plant progenies, and intercross, since they are cross-pollinated plants, intercross among them is allowed, okay, by random pollination. So they have their natural mechanism of random pollination, so they will have natural pollination, allow natural pollination for one generation, and which is known as intercross block. And in the next generation, what we'll have a composite of seeds which are formed by the intercross block is composited and grow in as a base population. Okay, so when the composited seeds from the intercross block is grown from the base population, it act again as a same base population, from where we are going to again select superior phenotype, and self pollination has to be done, and then seeds has to be harvested separately, and then the harvested seed has to be selected. Superior harvested seed has to be grown in an individual plant progenies, and intercross block for random mating or crossing among them are facilitated in intercross blocks. Okay, so this cycle is repeated once. It may be repeated once also, or it may be repeated several times also. It depends on the type of, it depends on the need of the plant breeder and need of the situation. So when the plant breeder gets the sufficient amount of genetic variation, he can stop or he can continue until and unless he gets the desirable genotype. Next is recurrent selection for general combining ability. In this method, selection is based on heterozygous tester cross performance, and it improves the general combining ability of a population. This method is used for genetic improvement of quantitative character. Remember this is usually for quantitative character, vital for quantitative character, and then it improves those characters governed by additive genetic variance. Okay, we have already studied this. Coming to the procedure of recurrent selection for general combining ability, first we have here several phenotypically superior plants are selected from the base population, and it is they are to cross with the heterozygous tester, broad genetic based tester. Okay, so when they are crossed with a broad genetic based tester, the replicated yield trials are conducted from the test cross seeds. Okay, when the replicated trials are conducted, superior lines from these replicated yield trials, okay, are identified and grown as a intercross block. Okay, so they are not going to directly select based on superior phenotypic character, but they are going to cross with a heterogeneous tester, and which is broad genetic based tester, and they are going to evaluate in replicated yield trials, and that they are going to, after evaluating superior lines out of it, then we are going to grow the lines in intercross blocks. Okay, so from the intercross blocks we are going to allow random matting in the intercross blocks by natural crossing. After that, the seeds are going to be composited. The seeds are going to be collected and grow in the next generation. From the next generation, it will act again as the base population like this, and we are going to again select superior plants, and we are going to select superior lines here. From the lines given here are red, we are going to select superior lines which are blue. Okay, and much like that of the earlier cases. So we are again going to cross with the heterogeneous tester, and we are going to evaluate the lines in replicated yield trials. Those which are superior, we are going to grow the individual progeny rows and form a intercross blocks. In intercross blocks, we are going to allow random pollination and so on. These cycles can be repeated again and again. Okay, so in this case, what we have done, GCA is evaluated, GCA is improved. Selection is based on general combining ability when there is a cross between the selected superior plant and the heterogeneous tester. Okay, remember that heterogeneous tester is broad genetic base and homogeneous tester is narrow genetic base tester. Okay, and heterogeneous tester is used for estimating the general combining ability, and homogeneous tester is used for estimating the specific combining ability. Next is recurrent selection for specific combining ability. Selection is based on homozygous tester cross performance. It improves the specific combining ability of population for a character. This method is used for genetic improvement of polygenic character. Okay, so in this, it is much similar to the procedure of recurrent selection for general combining ability. The only difference is the use of homogeneous tester instead of heterogeneous tester. So as we have already studied, homogeneous tester are narrow genetic based tester and this is to estimate the specific combining ability when we do replicated yield trials. Okay, next is reciprocal recurrent selection. In reciprocal recurrent selection, two heterozygous testers are used for crossing. So two populations are used, A and B. They will act as tester for one another. This is usually proposed by Comstock, Robinson and Harvey in 1949, and it improves both GCA and SCA of population for character. Okay, these founders are usually important for competitive point of view, and also so does the concepts also, and it improves the GCA and SCA of a population. So in this reciprocal recurrent selection, we are not going to improve GCA alone or SCA alone. We are going to improve both GCA and SCA. So this method is used for genetic improvement of polygenic character. Okay, and this method is also known as recurrent reciprocal half sieve selection. So we are not going to select based on full sieve here. So as we have already stated there, okay, we are going to have two populations A and B, which are both heterogeneous testers. Okay, both are going to have heterogeneous. So in this case, they are going to act as a tester for another. So first thing is we have to cross, and the test cross progenies is going to be evaluated in replicated yield trials. Okay, so coming coming to the first step. Okay, so the heterogeneous tester A is going to cross with another parent which is heterogeneous, which will act as tester for A parent. When we cross these parents, we are going to get the result. Okay, so the results, the test cross results are going to be evaluated in replicated yield trial. Likewise here, the parent for the population B, the A population will act as the tester, okay, heterogeneous tester. So when we cross the population B with heterogeneous tester A, so we are going to get test cross products. Okay, so the test cross product are going to be evaluated again here in replicated yield trials, and the replicated yield trials are not going to directly go for inter-cross blocks. Okay, it will be for those genotypes. The inter-cross row blocks will be for those genotypes only, and only for those which are superior in these replicated yield trials, which are found to be superior in these replicated yield trials. So what we have to do, the selected lines, okay, which are selfed and harvested separately, okay, and then with those lines which are found to be significantly superior here are only grown as individual plant progenies here, okay, and allow random pollination among them. So these intercross row blocks are composited and form a base population, okay, from which we can start again A and B. Okay, so these composites can form again a new population. Say for example A dash. Okay, another these lines can again form another by compositing these the seeds of intercross blocks. We can form B dash. Okay, and then this can form another base population, much like that of A and B, and then start the intermatting and reselection process again and again. Okay, so likewise here also the process, the continuous cycle of recurrent selection can be done until and unless the farmer or the breeder finds a desirable character. Okay, so some of the merits of recurrent selection are: it is efficient breeding method for increasing the frequency of superior genes in a population. And the next thing is it helps in maintaining the genetic variability, high genetic variability in a population due to repeated intermating of heterogeneous population. And next thing is it is made for selection is made based on the test cross performance, and only selected plants are allowed for intermating only. Some of the demerits here are this method involves loss of selection, so only selected plants undergo intercross blocks. So those which are not, those which doesn't pass through test cross, so they are rejected. Okay, crossing and selfing work. So this method permits selfing, which leads to genetic loss of genetic variability. Okay, recurrent selection, selfing is done. So when the selfing is done in cross-pollinated crop plants, they usually suffer from inbreeding depression and thus loss of genetic variation when they do so. Okay, so when they don't utilize hybridization or intercrossing between them, so they lost several genetic variations. And next thing is, it is not used directly for development of new varieties. Okay, this is a population improvement method from which we can there is a scope of selection of a suitable inbred line for hybrid breeding program, or it may be a base population for improvement. It cannot directly release as a variety. Recurrent selection cannot directly release as a variety. It is a base population from which a suitable parent can be selected in any hybridization program. Next thing is synthetic and composite varieties. What are synthetic varieties? It is a variety developed by selecting a number of inbred lines with good general combining ability. So this is a composite of this is a breeding method in which from the base population, collection of superior lines which are having phenotypically superior lines which are having good general combining ability. Okay, and after selecting of such line, intercross is going to happen. Okay, and we are going to mix the seed which are produced by intercross, and we are going to harvest the seed out of it, and that can be called as synthetic variety. Once synthesize the synthetic varieties can be maintained by open pollination in isolation. Why open pollination in isolation? We have to allow random pollination among the selected plants, because it keeps on increasing the broad genetic base. It keeps on increasing the variability, and at the same time it broadens the genetic base. Okay, and when it comes to cross-pollinated crop plants, these open pollination, the process of open pollination has to be done in isolation. Isolation distance has to be maintained, or any time isolation can be maintained, so that it is not, the genetic variation is not disturbed by any genetic variation of any other variety which is existing nearby the surrounding areas. Okay, features of synthetic varieties. Some of the features are, it is heterogeneous. Okay, the population is heterogeneous, not like that of pure line. In pure line, all the individuals are alike. So the seeds which are produced by the plants which are produced by heterogeneous population. Synthetic varieties are all heterogeneous. Okay, a synthetic variety can be developed by using clones or inbred lines or open pollinated variety lines. Okay, and in this case cross pollination is a must. Okay, because without natural cross pollination there is no improvement in the population, and it is maintained by again open pollination. Once developed, the synthetic variety has to undergo open pollination in isolation. Unlike composite variety, okay, exact reconstitution of synthetic variety is possible. So since we have the parents, okay, known parents in synthetic varieties which are of known general combining ability, so we can reconstitute, we can develop the same type of synthetic varieties which was developed earlier. Okay, and which is not possible in another type of variety, which is composite variety. And next is more adaptive to varying growing conditions as compared to hybrids, because they are heterogeneous population first of all, and heterogeneous population has a character which is broad genetic base. Okay, since they have a genetic constitution of all the composite parents, so they will have broad genetic base, and they will have a wider genetic base, and they can resist disease, biotic and abiotic stresses. Okay, So they are less uniform as compared to hybrids. Okay, this is the only demerit that they have. Okay, they are not uniform, and it doesn't look attractive in the market. Now in the next slide we have schematic diagram representing the procedure of synthetic variety. First of all, the superior lines are going to be selected, which is based on inbred line, which has to be inbred line, clones, or open pollinated varieties, and they have to be maintained as inbreds or clones. Okay, and then crossing among them is going to do based on their general combining ability estimation, and for that crossing block is maintained. Crossing block is maintained to produce synthetic 1 seeds, and those from these crossing blocks, estimation of general combining ability is done by half sheaf selections. So in this case, as an example, D is taken as common parent in both and estimate the general combining ability. So we are going to select if the general combining ability is high, we are going to select the same composite random mating block and recommend for commercial seed production, and this cycle can be repeated for production of synthetic 2, synthetic 3, and so on. Coming to two generations, synthetic 1 and 2. Okay, so these are generations which is developed in the second cycle of synthetic variety. So this synthetic 2 cycle generation usually lacks in performance compared to synthetic 1, usually because of heterozygosity as a result of random mating, which is allowed in the crossing blocks. Okay, and some of the factors which determines the performance of a synthetic variety are the number of inbred lines that are involved and the differences in the yielding ability of synthetic 1 and the base parents. Okay, so here, with keeping this in mind, Sewell Wright in 1922 has formulated a formula for the estimation of the performance of synthetic 2 variety from synthetic 1 variety before the generation of synthetic 2 varieties. Okay, what is synthetic knot variety? Synthetic knot variety is the base population from which we are going to form synthetic 1 generations. So synthetic 2 generation is syn 1 into differences in yielding ability of synthetic 1 and synthetic knot divided by number of parental lines. Okay, so from this we can formulate three factors for increasing the performance of synthetic 2. One is by increasing the number of lines, another is by increasing the performance of synthetic 1 lines, and third one is by improving the performance of the parental lines. Next is composite variety, much similar to synthetic variety. It is a variety produced by mixing seeds of several phenotypically outstanding lines and encourage open pollination to produce crosses in all combinations. And these lines are the lines used to produce the composite varieties are rarely tested for combining ability. So no GCA test and no SCA test is done. So we are directly going to select the superior lines and we are going to composite and form the random pollinate among them and select those as composite varieties. Okay, the yield cannot be predicted as there is no estimation of synthetic 1, there is no estimate of composite 1 or composite 2. Much like synthetic varieties, composites once developed, they are maintained by open pollination in isolation, because they are cross-pollinated crops. They are produced from cross-pollinated crops, so they have a higher chances of genetic admixtures from foreign pollens which are coming from the nearby varieties or weed species. Okay, so you need to isolate your hybrid seed production plot from away from other varieties which are crossable. Okay, so coming to the end of the slide, if you have any doubts you can ask